learn about the topic transposons, which comes under the recombinant DNA technology. So let's know about what exactly the transposons are. Transposons are also known as the jumping gene, the controlling element, the mobile genetics element, that is MGE, the transposable element, that is the TE. Mostly, jumping gene is used as an alternative name for the transposons. Now let us know what transposons exactly are. The transposons are the DNA sequences that can move from one location on the genome to another. In this red segment is known as transposon. And why is it so? Because this red segment it shifted or is jumped from one location to another location. Here you can easily see that this red segment was firstly present on this location and now it is here. So this red segment is known as transposons. Now you need to know about the various key points which are example and related to the transposons. It was discovered by the American scientist Barbara McClintock. Transposons were firstly discovered in corn, that is maize, during 1940s. She also got Nobel Prize in Physiology or Medicine in 1983. That is Barbara McClintock, who discovered the transposons, got this Nobel Prize also. And yes, you need to remember this enzyme very well. It is often asked in various exams also. The transposase enzyme binds to the end of the transposon and catalyzing the movement to another part of genome in the cut and paste mechanism. And what is the cut and paste mechanism? We will know further in this video. Now let us know about the types of transposons. Transposons are basically of two types, that is class 1TE and class 2TE. The class 1TE is also known as retrotransposon and class 2TE is also known as the DNA transposons. Class 1TE, in this the RNA are used as an intermediate form, and in class 2 the DNA is used as the intermediate form. In class 1 this transposons are found in eukaryotes only, but in case of class 2 it is found in both, that is prokaryotes and eukaryotes. Now let us know about the working of transposons. It works in two modes of transposition, that is first one is the replicative mode and the second one is the non-replicative mode of transposition. First, the replicative mode of transposition, it is known as the copy and paste mechanism. And why is it so named? Because it copy and paste its identity to another location also. Here you can copy and paste its identity to another location also. Here you can easily see that the transposon was firstly present here and now it is present here also. That is, it leaves its copy behind. You can easily see that yes, the transposon are present here also and here also. So hence it is known as the copy and paste mechanism. While in case of non-replicative mode of transposition, it is known as the cut and paste mechanism. See, it was firstly present here, but it is now present here. But its location which was here is not there. That is, it cut and paste is followed and it leaves no of its copy behind. It is not present here. So this was about the non-replicative mode of transposition. And yes, the transposons jump from one genomic place to another. It seems to be normal. But here comes the reason what effect it can possess. Yes, often it comes in mind, yes, what is the major thing that one segment is shifting from another location? So what can it cause? Basically it can cause a mutation. This can result in the disruption of genomic sequences also. And yes, remember why this effect is not shown? This is the major reason. About 50% of the human body is made up of transposable element, but they are silenced by different epigenetic mechanism, that is histone modification, DNA methylation. And yes, this is the reason that 50% of the human body is made of transposable element. but the mutation is not shown in such rate because it is silenced. Hope this is clear to you. Now let us know about the functions of transposons. The basic function is to move the segment of DNA from one place to another. The second one is to introduce a piece of foreign DNA into a genome, and yes, it play a major role in the evolution also. Let us know about the importance of transposon. It helps in studying the diseases, to develop the therapeutic practices for genetic disorder, to identify the unknown genes, to research of cancer, and yes, the cancer therapies. Now let us know about the various examples where this concept are found. In maize, in oxytricha, that is pond microorganism, the Drosophila melanogaster, various bacteria, human, and helitron found in eukaryotes. Now let us learn the various questions that are asked in various exams. The first one is the transposon that can be used during which one of the following. So various options are there: gene sequencing, PCR, gene silencing, and autoradiography. So the correct one is the gene silencing. Okay. The second one is the enzyme that catalyzes the transposition of an IS element is called. I told you already that it is. Here you can see that transposase enzyme. Okay. So transposase is the correct answer. Now it is saying that pick the odd one out: introns, cassets, jumping genes, and insertion sequence. So the answer is intron. Why? Because cassette, jumping genes, and insertion sequence, these all are the TEs, that is transposable element. Okay. On the mechanism of movement of transposable element, they are of two types. Okay. As I told, it is replicative and non-replicative. Okay. So you can take a screenshot of the notes if you wish. Hope this video is clear to you. Thanks for listening to me and have a wonderful day.